Hi, everybody. Welcome to the Bioexcel webinar. 83 now. Yeah, that is a nice number. I like it. Okay, today we have Elisa Fadda for the University of Southampton, and she will speak about restoring protein glycosylation with a GlycoShape. I'm hosting this webinar. I'm Alessandra, and together with Otto and also Richard Norman. And we are all from Bioexcel, the Center of Excellence for Computational Biomolecular Research. This webinar will be recorded. And during the webinar, you have the option to ask questions, so all the time, and we will read those questions at the end of the webinar. So if you have a microphone, I will unmute you. Otherwise, I will read the question in place of you for Elisa. And so to type your question, you can use the Q&A function that you find on the bottom of the Zoom application. Depending on which operating system you have, you can see this symbol or this symbol. Please specify if you don't have a microphone, so I know in advance. Thank you. After the webinar, maybe you still have some question. There will be a possibility to ask a further question within 1 week to Elisa using the Bioexcel forum. That's just you go to the forum and there is an entry with the webinar number, and then you can just ask your question to Elisa. Something about Elisa. Elisa is currently an associate professor in the pharmacology in the School of Biology Science of the University of Southampton in UK. Her career started in 2004. She was a PhD student in chemistry at Université de Montréal in the group of Dennis Salo-Hupps. I am not sure I pronounce his name correctly, and developing DFT-based methods for the prediction and NMR shielding. Then she moved on and she worked at the hospital for sick children in Toronto on biomolecular simulation of ion and proton translocation events through transmembrane channel and proton pumps. Then she had a period between 2008 and 2013 at the University of Glasgow, where she was start to study glycoprotein and carbohydrate active enzyme using still molecular modeling. And in 2013, she became associate professor of the Computational Glycoscience Research Group and the Department of Chemistry and Hamilton Institute at the Maynooth University in Ireland. And then in 2004, she became associate professor in pharmacology at the School of Biological Science and Biological Science. Now she will speak. I will give her the word and she will introduce her work to us. Thank you very much. I just close. Sorry. Please.
Can you see my screen? Hello?
Yes, perfect.
Yes. Thank you. Hi, everyone. Thank you, Alessandra, Otto, and Norman for inviting me today to talk to you about glycans or carbohydrates, if you haven't heard the word yet. Sorry, I'm just minimizing myself. There. Okay. So, and how, like, I'm going to show you how to include them in your studies and why, especially that it is extremely important to include glycosylation in your study and how you can do it quite easily with a set of tools that we are developing called GlycoShape. So if you haven't, if you are not let's say like familiar with carbohydrates or the functional carbohydrates in biology and think that carbohydrates pretty much are related to food, you're not alone. Like, so most, if you look at, for example, a search on Google that I updated yesterday with all the aid of this very smart, or apparently should be smart, AI tools, the word carbohydrates is fundamentally associated with food and with any type of food that contain carbohydrates. You might find some role of carbohydrates, presence or absence in diet. You should eat carbohydrate or not eat carbohydrate, and so on. But if you look at the impact of carbohydrate that is not food related, so non-dietary carbohydrates in health and disease, that goes very, very low at the below, like at the very bottom of the search box, if you can actually find anything about it. So the point is, like, the carbohydrates are actually the most abundant biopolymer in nature, and so their food, like their role in food, is just one of many. So possibly the knowledge that carbohydrates are fundamental in health and disease, and they're not just in your bread or potatoes, came more to the public, let's say, like to the public level, of the public knowledge, rather, during the COVID-19 pandemic, where, like, you can see from this image, like, this is a SARS-CoV-2 variant, like, with like this kind of like corona around, and that's what is called the coronavirus, which are like the spike proteins, so the like the fusion proteins, which are covered actually in carbohydrates. This is a magnificent reconstruction of the old variant, like from Romy Amara's lab at UCSD, and you can see all the blue that is used to represent explicitly in a computer model the glycans, or the sugars that cover the spike protein. So actually what people realized, and they didn't know before, is that in terms of when we talk about virus, the sugars are everywhere, to paraphrase a poem. So we have to include viruses, like sugars, when we talk about viruses, but and we not only talk about it, but when we study viral infection. But that doesn't really, let's say, like pertain to viral glycobiology, but it pertains to any domain of biology. In fact, like, I found this like this image when I was looking to, like, say, for some picture to show you the glycocalyx, which is like say the layer of sugars that surrounds all the cells and the epithelium of our in our bodies. So, and this is particularly, I thought impressive, because it really, like, shows you color, how, like, cells in this case are covered by sugars. So and in the left, there's the cells that have sugars on. In particular, these are mucins. They are like bound to a fluorophore, and in the absence, which is on the right. So if you think about it, anything that concerns cells, or if you're studying any interactions with cells and other proteins, requires the like having sugars or considering sugars. So, but if you think, like, the impact of sugars in human biology is mind-boggling. So, like, if you think that approximately like 4%, so between 3 and 4% of the human genomes encodes for proteins that are required for cellular glycosylation, for glycomodification and degradation alone. So you would think that this is quite an investment in evolution, and such an investment is not really, or doesn't make any sense if it was dedicated to such a simple decoration. So glycans are far more important than decoration, and their variety and diversity in terms of structure and sequence is mind-boggling. Considering the glycans can also be branched rather than linear peptide, linear polymers such as proteins or nucleic acids. So the variety is mind-boggling, and this variety is essential to their mind-boggling amount of functions that they do in our biology. So what do we know about this? Well, we know very little in fairness. So, and this mystery that surrounds glycans and glycosylation was never put in better words than by Ajit Varkey and Stuart Kornfeld, who are like, the trailblazers of glycobiology and the fundamental book of glycobiology, which is, by the way, available for free online. So in chapter 1, they actually describe that glycans can be considered as the dark matter of the biological universe. And this is quite true. Like, it's just like we know that they exist and they're irreplaceable and their function is essential. However, we don't know how they do a lot of those function, how they execute a lot of those function, and what is the fundamental units that do execute those functions. So why is that? Like, a lot of the mystery is due to the fact that the glycans have a chemical structure that renders them intrinsically disordered. Like, if some of you have worked with intrinsically disordered proteins or attended any intrinsically disordered protein talk, you've seen pictures like this, where, like in this particular picture, like, which has like an exposure time of 5 seconds, so it's overexposed. There's like, you can see like there's some people stationary, but like in large part it looks like a deserted street. However, it's like a street in New York, and for those of you who have been to New York or live in New York, it's quite rare to have a street that is completely empty. Indeed, everybody it's like the street is full of people and everybody is walking quite fast. Therefore, you cannot capture their image, but like you can see some blurry like distribution of colors here and there, the signal the probable presence of somebody, but you cannot see really what these things are. And this is actually what one of the reasons why glycans are and glycan function are still largely mysterious. So we can't see them. So like in terms of structural biology, glycans, so part of them can be only resolved when they are restrained, when they're bound. So, and the point is like that you can see part of the glycan that is actually attached or linked, or let's say, restrained by a protein on a binding site, for example, but you cannot see the whole glycan. So like that part is not generally the whole glycan unless it's an oligosaccharide, but is a part of a much larger structure that you need to reconstruct to understand the biology associated with that event. So in case of cryo-EM, like the actual issue is actually way more involved. Like you not only have an intrinsically disordered structure to deal with, but also a very heterogeneous subject. So our glycans are not DNA templated, so like there's no part of the genome that tells you what glycans goes where. So there's their glycosylation site that are occupied and glycosylation site that might not be as occupied, and how that is chosen or selected is very difficult to predict. So not being templated means that at every glycosylation site, there is a different level of heterogeneity. So one glycan can be in a glycosylation site, and in the same protein, that glycosylation site could be occupied by a different glycan, or slightly different, or widely different. It depends on the case scenario that you have. So because of how you collect and resolve the images, cryo-EM images are averaged, so are results of averaging. So like your glycan, or the glycan density that you find, is actually an average of a lot of different structures of that exist on those proteins. So what you see generally is the root of a glycan. Could be like, for example, one GLUCNAC for N glycan, or one GalNAc for an O glycan. If you have, those are the most common modifications that you might have. And that is not the glycan. That is just a very small piece of it. It's the only part that generally is so static and commonly shared by all this heterogeneous species. So what do we do to understand, like to overcome this problem in structural biology? And we know that structural biology is a fundamental part of or portion of for us to understand, or side, let's say, part of area of biology that allows us to understand function. So we use in my lab computer simulations. In particular, we have reached a technological advancement and sophistication of the science underlying this simulations that allows us to really explore cellular, like events at the cellular level, the molecular level on a cell surface in particular, and to compare them with like experimental biology. And this is extremely important when you have a topic or you're studying glycobiology, because, as I said, there's no template, there's no DNA that tells you what you should, like what you're looking at. Like, so you need constant collaboration and with many experimentalists and vice versa. So like in glycobiology, we say it takes a village to write a paper, and that's usually the case, which is also a pleasure. So glycans mediate every possible interactions of the cell and proteins interaction that I can think of. So and that's why I like to image glycans just to make people understand what is like little ends sticking out from the cell surface, from the the surface of secreted protein, of transmembrane protein. They're all imaged here in this rendering, that extend out with all different fingers, like if you can imagine different types of fingers, waiting to shake a hand and to transmit a signal or forward a particular process that determines life in health and disease situation. So these handshakes can be the basis of an interaction with lectins, or lectins are actually proteins that bind carbohydrates, but don't do any chemistry. Antibodies, or cases, which are like enzymes, so that do chemistry. In particular, that this acronym refers to carbohydrate active enzymes. So they are cataloged, like you can find the cases database online. So glycans are essential for folding and for structure stability of secreted proteins and transmembrane proteins. So like this is intrinsic in the biosynthesis of glycan and the folding of the protein. So they need to be replaced to have a stable protein. So, and actually all these interactions replaced in your simulation. All these interactions are actually exploited by toxins, bacteria, and viruses as a first port of entry for infection. And in particular, an example that pretty much is emblematic to me of the role of glycan structure in particular in a pathogen infection is given by flu. So this handshake is actually why you get a flu infection, and this is particularly important nowadays. We're talking about this new infectious avian flu or bird flu that like has potentially found a mutation that brings it closer to infecting human more easily. And we have seen actually cases of humans being infected by this new strain of avian flu. And avian flu actually recognizes or enters the cell by recognizing handshaking a carbohydrates hand, which is in particular, the blue one represented here is common in the respiratory tract of birds. Like you can see, is a straight out pointing straight like sialic acid, which is like the most, let's say, stable structure of an alpha-2,3 linked sialic acid. Meanwhile, the human upper respiratory tract contains more commonly glycan that terminate with a 2,6 sialic acid, alpha-2,6 sialic acid that is actually bent. So, or as is most populated conformer is actually bent. So the recognition of these two structures like differentiates an avian flu from, in particular, hemagglutinin from a human flu. So like this gives really an idea of how this recognition of a glycan can progress into a disease state. So I can do a lot of simulation. I've done it all my life, but not everybody, especially in glycobiology, is either willing or able to run simulations to understand their structural biology problem. So we decided like many years ago to use our simulations and to store them and to analyze or pre-analyze them to provide service for the community, like so not only the glyco community, but actually to make glycan more accessible and usable for everybody. So to have people, including like glycans, they are in nature more often. And in order to do this, we developed a service that's called, or like a web-based tool that's called GlycoShape, which is like a structural database, but also a glycobiology toolbox that allows all of you to, as easily as possible, include a glycobiology site to your work. So now we have like but last time I checked, about like 639 unique glycans between like full forms and epitopes from the human glycome, and mostly from the human glycome, with examples from all species. So this variety and diversity allows you pretty much to build any human glycoprotein that you want with GlycoShape. So we like if there is a glycan that we don't have, but that you want, that possibly from a model organism that we haven't explored thoroughly, please let us know and we'll build that glycan for you. So this, like the tool that you can see here, like on the top menu, like it's Reglyco, is a bespoke tool that allows you to link those glycans in the database to your protein, and pretty much is highly customizable, so that you can link whatever glycans you want to whatever glycosylation site should be accessible to that glycan. So this work is pretty much the overarching mission of anybody in my lab. So everybody has his own project, but everybody shares the GlycoShape project. So everybody makes glycans and analyzes them and puts them in the database. The heavy lifting in terms of the design of the database and the design of all the tools in the database, it still continues, is primarily from Callum and Ojas, who are here highlighted here. The paper is being published recently for all details that I might skip for time constraints. So GlycoShape has been out now quite a long time, we're not very old, but we launched the website in the end of 2023, and since then it's been picked up quite readily. So like it's used all over the world. This is a map that we built like around October, and all the dots represent different IPs that we collect when you connect. Like we don't ask you to sign up for anything. We just collect your IP to build these maps, so to see how and to monitor how GlycoShape is used all around the world. So we have like on average and consistently since the beginning of 2024, an average of 10 people glycosylating the proteins every day. And that doesn't really, we're not counting us ourselves, which is like what we do all the time. So like this could be you, like if so like that's if you, especially if you live in some places we don't have, please do log in. If you have your city, please do log in as well. We're gonna have bigger dots. So for those of you that are interested in how do we transform our MD trajectories into data, into structures, in particular, how do we analyze the trajectories and collect the highest populated conformers that you can use to rebuild your protein? This is done by a bespoke protocol that we call the glycan analysis pipeline. So we build or we simulate our glycans through a kind of a grid search inspired framework where we use the glycosidic linkage's main degrees of freedom to build microsecond cumulative sampling. So this is quite thorough sampling for all glycans that we have. It doesn't matter how small they are or how large they are. So we collect this cumulative, this data, all these different and independent runs into a cumulative trajectory that we transform into a distance matrix. So the lower half of this matrix is flattened into an array, and then we run PCA to achieve dimensionality reduction. And then the clustering analysis is done with a Gaussian mixture model through on the PCA. So the representative structures from this clustering is given by the KD max of this distribution. So you can have usually for every glycan you have up to 5 if the glycan is very flexible. If it's less flexible, you might have less than 5. But like 3, like generally between 3 and 5 is the happy medium. And each glycan in the database is identified by what we call a glytoucan, what is known as glytoucan ID. So as you know, each protein in the PDB database is recognized or is identified by a PDB ID. This is the glytoucan ID is the equivalent of the PDB ID for glycans. So it's a much bigger code because there's a lot of different type of glycans, like linkages and symmetry. So it's a bit bigger. So but this is pretty much how glycans are identified. But if you want to search a glycan by UPAC or by VERX or by any other methods, we have implemented as many as we possibly could. We also annotated available biological and chemical information, and this is actually growing nowadays through integration with existing glycobioinformatics resources such as Glycosmos and Glygen, sorry. So how does reglyco work? If you have obviously you are in front of a computer, but if you want to give it a go, minimize me and give it a go like you can do while I speak. So in this case, I'm just illustrating one of the, say, less common example. So because everybody has a protein structure, but not every protein structure has an associated list of glycosylation sites that are occupied, and in particular, a list of what glycans goes in what glycosylation site. So because the last two of these conditions, like this is the realm of proteomics and glycomics. So not everybody has this information at hand, but if you do, and usually in UniProt, like you will find like under PTMs, if there are any studies related to the glycosylation of that proteins, I invite you to follow that information closely. So provided that you have a protein structure, a list of glycosylation site, and know what glycan goes where, you can actually use reglyco by fetching the PDB ID, like if you have a PDB, your protein is in the PDB, or UniProt ID if you want an AlphaFold, AlphaFold 2 from the EBI EMBL database, or if you have your own protein, which is most of us do, like, because you made some modification or because you have it on your desktop, you can upload it your own into reglyco through PDB format. So the glycans that you choose, it could be any glycan, like if you have a list of glycosylation site and the list of glycans will invite you to choose those. So the glycans are linked to the protein with a genetic algorithm that minimizes a loss function. So it's like a repulsive potential. So accounting for steric hindrance, we are implementing at this time an attractive potential, but it's not in reglyco yet. And most of the time, you really don't need it in our case. So the choice of linkage is extremely important because it's actually like direction of the side chain in this case, like an asparagine in the case of an N-glycan or a serine or threonine in the case of an O-glycan. Most of the time gives the direction of the O-glycan in space, like could be directed towards a particular side of the protein or another side. That obviously depends on the resolution your protein is at. But so the choice of an appropriate linkage is extremely important. And we use for N-glycans the crystallography database known as Privateer from John Agiri's lab in the University of York. And for O-glycans, we have developed our own MD database that covers any O-glycan and C-mannose glycan. So that's quite precise. So based on that, the minimizer will select which of those clusters available for each glycan fits best in order of their relative population, and we will put it in on your structure, as you can see here. So the choice of cluster max in this case is second glycan. So you can also rebuild in one shot if you trust the annotation in UniProt, and it's not always available, but in some cases it is available. So we like to see, be quite careful about this. Usually annotation of UniProt glycosylation is done by sequence. So this is the most common case that you have. So you got the protein structure, and at this day and age, who doesn't? Because you might not have a PDB, but you definitely have an AlphaFold structure. Good or bad, I'm not the judge of that. You'll be the judge of that. But you might not have a list of glycosylation sites, and you don't know what glycan goes where. So this is usually 90% of the cases. We can help you for N-glycan through a protocol known as GlucoNAc scanning. In this case, the GlucoNAc scanning uses the power of reglyco to assess if a glycosylation site is accessible by trying to fit a GlucoNAc. So in this case of this small protein, for example, you have two sequences that could be glycosylated, only one is occupied, and the other one doesn't fit because it's an internal sequence. It's just like this two, there's no space for a glycan in between these two helices. So only one is present here, and then you can continue and functionalize that glycosylation site with the glycan that you want. And for all glycans, you will have to decide if you want to put it or not, and where. And that's for all glycans. Actually, it's fundamental that you have glycobiologists helping you, suggesting if that glycan is in that place is correct or not. So this is all I said. The GlucoNAc scanning actually is extremely powerful and very accurate, and it scoring 93% against experimentally profiled structures in the PDB. So we have a really high rate of success in this case, and this stems from the actual biosynthesis of N-glycans and the actual accuracy of reglyco and of the glycosidic linkages. So I'm going to show you how this works on an example, and how this, in particular, this process can help assessing the glycosylation site accuracy in all structures, because you need to be critical a bit about what structural information you get. It might not be optimal for glycosylation. I mean, you need to actually adapt it. So this is CD16B, which is an Fc gamma receptor. There's a lot of data on it, structural data and glycosylation data, because of its extremely important role in immunology. So we know that we chose this particular structure here, and we know from experiment done in neutrophil bound CD16B, so membrane bound protein, that these are the sites that are occupied and these are the sites that are unoccupied. If you screen this with reglyco, actually, this is it gives you 2 false negative and 2 false positive, which is the worst results that we've ever had with reglyco. But there's a reason for that. So we'll start with the 2 false positive. So actually, those 2 false positive are only false if you consider a neutrophil bound CD16B. If you have a CD16B that is recombinantly expressed, so only the soluble part, which corresponds to this, or to the structure that you submitted to reglyco, both of these are actually occupied. So, and this is actually important to understand that glycosylation, it's really dependent on where the protein is in its natural environment. So if you are simulating, let's say, a recombinant protein versus a membrane protein, the glycosylation might change radically. So in the case of the false negative, then you have something that you can work with. So for example, the blue or the cyan structure indicates it's resolved with a wrong orientation of the N and O atoms relative to the structure where the glycosylation occurs. That is the pink one that actually was resolved with glycans attached. So you can see that the nitrogen is actually it's orientated towards the outside of the protein. So you can do this change by a button that's called swap, that comes up when you have a clash in reglyco. So you can swap the coordinates of N and O between each other, and it generally solves the issue. So in the case of this N74, you have a clash due to the fact that this area is actually highly disordered and is being resolved. Well, it shouldn't have possibly been resolved at that resolution. So you have extremely mobile coils. So you see this asparagine has been resolved, pointing towards another loop, and it can be absolutely rotated out. So we have no rotamer search, automated rotamer search in reglyco, because actually we prefer not to have everybody changing the structure of proteins, but only to do it if they're knowing what to do. So this is actually the end of the fully glycosylated neutrophil bound CD16B, according to the analytics from Manfred Wörl's lab. So this to show you that you can do extremely more difficult projects. For example, there is nothing that is more densely glycosylated HIV GP120, and this is the work published recently by Joe Allen and Max Crispin, colleagues of mine in University of Southampton, where they followed, or they used their glycoproteomics profile and reglyco to rebuild the envelope protein in HIV. So what about AlphaFold? It's so in the terms of AlphaFold, we discussed in collaboration with John Aguirre at the University of York, the possibility that actually AlphaFold structure, or ML predicted structure, were ready for glycosylation. And this is actually true, in the sense that a deep neural network that is trained on protein structure that do carry cofactor and PTMs should predict structure that match those predictions. So it should implicitly know how to build or to fold a protein, even in the explicit absence of these modifications. So the high PLDT alpha-fold structure should be glycosylation ready, and this is actually what we argued there are with some example. But in our recent paper, we extended quite extensively this test up to 12,000 over like almost 13,000 and glycosylation site, and we see that that is actually true. So we have that most of these proteins are glycosylation ready, like this is AlphaFold 2. And most of the issues like with fitting can be solved by reglyco alone across the PLDT scores, except for extremely highly resolved structures or very poorly resolved, like predicted rather structures. So this is most of the clashes actually shown by this example from exostosin protein, where we have from cryo and X-ray crystallography and lots of AlphaFold structure through AlphaFold 2 and collab-fold. Then you can have, at the time of the publication, we didn't have AlphaFold 3, but you can use AlphaFold 3 so the clashes are only due to rotamer position of this asparagine that, having 5 structures in AlphaFold 3 or collab-fold 5 structure will solve it immediately. So there's no problem. So why should I rebuild the glycans if I add a protein? Well, if you don't, you'll miss half of the whole story. So that's generally my experience. So, and I can give you different examples. So while reconstruction in 3D structural glycans provides actually an accurate description of the native and functional form of glycoproteins, and nothing is more evident than when you have a very densely glycosylated spike protein, as you can see here, where the right-hand side structure doesn't even exist. It's not biologically relevant at all, and it would not exist. So, but also, glycans might be doing all the heavy lifting, like so in the process that you're trying to simulate. So the presence of the glycan not only it's not decorative; it's actually functionally it's functionally essential. So in this case, actually, the glycan is doing the whole protein-protein interaction. There's like it's just mediating like the interaction with the protein. So, or like the structure of the glycan actually can explain how there's some events occur and what are the triggers, how a protein can orientate in space because of the structure of the glycan it's recognizing. But now, today, I'm just gonna spend the last possibly couple of minutes talking to you about like how glycans mediate this protein-protein interaction. This is completely new, just published a couple of days ago, weeks ago, I don't remember, like not weeks ago, maximum 2 weeks. And in particular, showing you about or an example about how glycans pretty much are like kings, like if you want to allow me the word, of anything that happens in immunology. So glycoimmunology, and in particular, the interactions with Siglecs, which are sialic acid recognizing lectins. So they mediate immune response. Like in particular, like all these Siglecs are human Siglecs. You have like 2 different groups, and they are like massive diversity, and they're all tools that are similar. They look similar, so they have like similar domains and with different number of these green domains, but the same number of the one top domain that recognizes sialic acid. But they all recognize sialic acid in different contexts. So they're very, very, very specific. So in particular, today I'm going to mention Siglec 10, which it's actually the starring role of this story, where Siglec 10 inhibits like inflammation by binding the secreted glycans, so like just like glycan secreted proteins, and polysialylated epitopes. So it particularly binds CD52. CD52 is a peptide. So it's a 12 amino acid, like, in this mature form, it's a 12 amino acid immunoregulatory glycopeptide, which is a sequence like this that binds this protein, that's called HMGB1, and then binds cyclic 10. So we were actually asked by our collaborators, Nikki Packer and Len Harrison in Australia, how this all takes place, how this CD52 works. And in particular, CD52 is very, very small, but it has very complex glycosylation. It has two types of glycans, one N-glycan and one O-glycan. So the N-glycan at N3 is actually in the active CD52 fraction is extremely sialylated. So it has a lot of sialic acid everywhere. So it's essential for, and you might think, yeah, because it binds cyclic, so of course it needs to be sialylated. But also as an O-glycan that has in its it's highly sialylated, it's hypersialylated in the active form. So the active form of CD52 is hypersialylated, and this is mass spec data, so extremely accurate mass spec data. HMGB1 is actually, for those of you that are interested in structure and structural prediction, is actually could be the nightmare of any structural prediction, because it has two domains, box A and box B. Their sequence is very, very different. There's there have like 25% identity between the two box domains, but the structure is almost identical. So like if I superimpose one to another, I get an RMSD of 1.2 angstrom. And the fun fact of this protein is that it binds everything. Like it's secreted and nuclear protein, and it binds p53 transactivation domain, the box A, CD52 secreted in a secreted form like box B, and it also binds DNA. So it binds pretty much anything. So the question was like, how CD52 regulates inflammation, suppression of inflammation, by binding HMGB1 and then binding cyclic 10 was like with no structural information, was quite a question. So what we did a lot of work. So this is all work from Silvia Andrea, like in my lab, that she dedicated extremely long time by simulating different glycosylated peptides in all different forms. And what we noticed is like the presence of sialic acid actually changes the structure of the peptide if we compare it to the same exact glycans but not sialylated. So you have like a more extended peptides in the presence of sialic acid. This is actually due to conformation around N and around S9. So in proximity of this of the glycosylation site, where all helicity is completely suppressed. You see, like there's quite a helical in the absence of sialic acid, but this helicity is completely suppressed in the presence of sialic acid, forming more extended structures. So what we did was like that we by analyzing how the glycans actually shape this peptide, we found that the peptide was shaped in a remarkably similar bend, like around this area here, as the p53 transactivation domain bound to box A. So we actually used this as a template to build a potential complex, and we ran the hell out of it by rebuilding the glycosylation again on top of it with GlycoShape. And what we found is like remarkably is that this O-glycan is actually what binds HMGB1, which is like here in blue. Meanwhile, the N-glycan position all the sialic acids, and together with also the HMGB1 glycan that is shown in teal color here, to the cyclic 10 for binding. So why, the reason this hypersialylation is also the reason why there's a preference of box B versus box A, because all the contact residues that this glycan makes with HMGB1 box B are not present except for one in box A. So that is automatically excludes box A from as a binder, as a receptor. Last like words of wisdom. So you're like, why should you glycosylate a protein? Well, because a protein is glycosylated, especially if it's secreted. 70% of secreted proteins are glycosylated. So think about it. If your protein is secreted, it's very, very good chance that it has glycans on it. So, and if it's not secreted, it might have ogloknak. So, like as a cytoplasmic post-translational modification. So you're not get rid of glycans, or you might not be safe. So, do you have any glycosylation sequence? So, N glycans, for example, follow a sequence. O glycans don't. So you need to have a glycoproteomics analysis. If you have data, please use that, the data in the literature. So, and then you can rebuild your glycoprotein quite readily with GlycoShape. So thank you all very much for your attention. If you have any questions, I'll be happy to answer. Thank you.
Thank you, Lisa. Great talk. Thank you very
Thank you.
much. We have a 3 question in the Q&A that they are without microphone, so I will read it. So the first one is, the glycosylation linkage involve dihedral angles?
Involved what, sorry?
Dihedral angles.
Yes. Yeah.
Yeah. Okay.
So it's yeah, like it depends on how many, like like what type of glycosylation linkage. So you have 2 or 3, a 6 linkage, or any other linkage. So
Okay.
at least two dihedral angles. Yeah.
Okay.
It's a glycosidic bond. Yeah.
Oh, thank you. And we have another question that, have you done any comparison of your protocol for including the glycosylate AA with the one from Charm GUI?
Sorry, I don't understand.
You can read it maybe. I also don't know what is glycosylate AA. With the one with
Amino acids. Okay, so
will be glycinate amino acid. Yes, sorry. Yeah. So it's the
yeah,
second
can you read it
question.
again? Sorry. Yeah.
Have you done any comparison with your protocol for including the glycosylate amino acid with the one from Charm GUI? Did you have
Okay,
any
so
comment?
yeah, so like the carbohydrate builders such as CharmGUI or glycom.org. So glycom, they build glycans based on crystallographic data. So they're built as a like using parameters for their glycosylated linkage derived from crystallography. So it is like, so usually those are dimers, like, so a disaccharide. So like you cannot see bigger glycans usually than that. So their data, they're obtained from disaccharide. So you're building a whole glycan from a disaccharide, so a sum of disaccharides. So this generates usually extremely extended structure. They're not necessarily the same as the structure of the glycan like in solution. So it might be very similar if you have a very small carbohydrate, like so like TN antigen or like very small O glycans. But for N glycans, like when you exceed let's say like a polymonocytic pentasaccharide, so like you have arms and flexible arms or different types of functionalization, the like the similarity between what you build with CharmGUI or Glycom and what you do obtain from simulation are completely divergent. Like so, it's just but like those tools are not made for simulating or representing equilibrium structure. It's like when you build a structure of a protein or a peptide using, I don't know, Schrödinger maestro, you would have an extended structure, like which doesn't represent the structure of that peptide at all. So you can see it a bit like that, but that tool was not made to represent equilibrium structure or equilibrium properties. So that's how we should think about it.
Yes, so I follow up a question that I jump one question just for people to understand. So I follow up with a question that is connected to this one.
Yeah.
How does sorry, how does your database different to other tools to build glycoprotein, example, Charm GUI? And is there an existing pipeline to get force field parameter from create a structure from your database?
Yeah. So as I said, like you know, like the Charm GUI and glycam.org, like so, or also the tool that now I don't remember the name, and I apologize for that, that was developed for GROMAX, uh, are carbohydrate builders, so are glycoprotein builders. So they're made for as a first step in order for you to run a molecular dynamics. So like they're never made to rebuild a glycoprotein and to look at it or to suss out if the glycan is interacting with something or not. So the reason why you should like when should you use one or another? If you're running molecular dynamics, I would say like you can use whichever, like it doesn't matter because you're running. The point is like the structure of the glycan from GlycoShape, it's already equilibrated. So it's very similar to what you will get if you do multi-microsecond simulation. So the point is like, or at least the analysis, the resulting of the analysis of those structures. We don't like what you get from from Reglyco when you download the protein is a PDB, because now like in the now second version of our toolbox, because I said we're old but not that old, like we had to cater for the majority of our users, which are structural biologists. Like so, like they don't they need a PDB structure where the atom types are PDB format. So you can upload like that structure, and any pipeline that is allows you to build a topology file would like that's compatible with it, like so perfectly compatible. Is like how can I use a structure from the PDB and associate parameters, either charm like or amber or whichever force field you want to use. So we will use we will or that at least that's the plan, have a little like script that allows you to produce uh, like charm topologies or amber topologies from our PDBs, but we don't have it yet. Like it's just a matter of changing atom names. However, glycans are not really portable, so it requires a bit of reading and a bit of thinking. So it's a master project student for like master student project for sure, that we haven't come around to do that. But yeah.
Oh, thank you very much. There is a final question, I think, that I take is more general one. Are the glycan helping in sequence alignment or superposition stability alignment? This question popped up when you were presenting on the last part of your talk, when you were showing some example. Just to put for you some context.
Oh, the guy, yeah. So like, yeah, thank you. So now I think I might understand the question. So the glycans actually that are on the CD16, sorry, the CD52, they select, because the binding is done by the glycan, it's not done by the protein at all. It's not done by the peptide at all. Actually, if you think about it, the glycans are actually much larger in terms of mass than the actual peptide. So there is the protein that is like, they should actually sit in the back seat in the sense that the driving is done by the glycan. And how the glycan is structured around the protein when is sialylated, forms contacts with the proteins, with a protein only of box B, not a box A, because box A doesn't have those attachment points. So it's like it's a protein like if you think about it in terms of protein interaction, the interaction occurs because you have fundamental contacts. So in this case, one of the parties is actually the glycan. So and that selects for box B rather than box A. They have identical structures, but very, very different sequences. So that's the important part. Like that, the reason why I presented that is to make you understand that if I didn't have the glycan, I would have missed your whole story, because the protein does not bind at all. Box B is the glycan that binds box B.
Okay, thank you very much, Elisa. I think we finish here with a question. I just want to announce, I just share my screen, I make mistakes, sorry. Share my screen to announce the following webinar. And there are still some questions, but I invite people to contact Elisa via forum or with other means as for the following question. Yeah. So just
Thank
give
you.
me a moment. Yeah, please go ahead. In the meantime, I share the screen. Yeah. Okay, so I want just so I just want to share with you the following webinar. So next week we will have Siverian Malik that he will speak perspective on simulating the whole cell with Martini. And then we have some week break, and then the 8th of April we will have Marco Giuliani that will speak about modeling antibody in a post-alpha fold area where we are now. And looking forward to meet you there. I thank you, Elisa, for the nice presentation, and all of you for the attention and for the nice discussion. Thank you. See you next time. Bye-bye.
Thank you.